Hi everyone, we're looking at a process known as meiosis in chapter 11. Meiosis is also a way that cells can divide, but it's a little bit different than the process of mitosis, which we talked about in chapter 10. And I'll try and highlight some of those key differences to you. For starters, I would like you to kind of think about some of these questions to remind you about some of the things we talked about in mitosis. So go ahead and pause the video and see if you can work through these, and then I will go through them, and it'll be just kind of a nice review. Okay, so the first question asks, how many of the following, for just a random skin cell, you just take a skin cell right off of your skin, that's what we'd call a somatic cell, meaning it's just a regular cell in your body, and that's going to be a diploid cell. Diploid means it has two sets of chromosomes, and this is going to be the case of all the cells in your body, save what are called the gametes, which are a different type of cell, which is what this whole chapter is about. But for starters, the chromosomes prior to the S phase, this is going to be 46 chromosomes. All of our body cells have 46 chromosomes, and this is because there are two sets of 23 chromosomes in there. And if you take 2 multiplied by 23, you get 46. The number of sister chromatids after the S phase, so after all of these have duplicated, then that would be double the number of chromosomes. So this would be 92 sister chromatids. And the homologous pairs of chromosomes, we'll assume this is talking about prior to the S phase. This is going to be 23, okay? Because you have two sets of 23 chromosomes, and the chromosome of one number on one set has what's called a homologous pair with the chromosome of the same number on a different set. Okay, so two sets of pairs, so 23 pairs for a total of 46 chromosomes. Okay, in mitosis, the steps look like this. There's prophase, there's prometaphase, metaphase, anaphase, telophase, and then lastly this process known as cytokinesis. Okay, so the big events that are happening in each of these: prophase, all of the chromosomes are condensing. Normally the chromosomes in the nucleus of the cell are kind of like spaghetti. They're sprawling out, makes everything more loose and easily accessible. Well, in prophase, those all condense, and also the nuclear envelope is going to start to fragment, as well as the membranes and some of the organelles like the endoplasmic reticulum and things like that. In prometaphase, the big event that's happening is that spindle fibers are emerging and attaching to this region on the chromosome known as the kinetochore. The kinetochore is just kind of this sticky part of what's known as the centromere. And recall, when a chromosome duplicates in the S phase of interphase, it looks kind of like this. And the place that it's attached is called the centromere. And then on there is a little kind of a sticky region where the mitotic spindle can attach to. And there's going to be one that's kind of on either side of the sister chromatids to pull them apart. Well, the attachment of this spindle to the kinetochores is what's happening in prometaphase. It's kind of the big event, what's going on there. And then in metaphase, what's happening is that all of those chromosomes are lining up. So these are all the duplicated chromosomes, so they're all going to have sister chromatids, which is what I'm kind of trying to illustrate here. They're all lined up in the middle, and then in anaphase, then keep in mind they're all attached to what's called a microtubule spindle. And then in anaphase, those spindles start to shorten, and it's going to pull the sister chromatids apart. And then once they've broken apart, they are now called individual chromosomes, and they will migrate to the different regions of the cell. And then telophase, what happens is that now there is a cell which is kind of getting a little bit longer, and those chromosomes will be kind of localized in different poles of the cell. And then the nuclear envelope is going to reform to form kind of a two nuclei structure within the same cell. And then the last step is cytokinesis. That's going to be the formation of a cleavage furrow, which is a ring of actin filaments that kind of surrounds the cell and basically pinches it shut. So that is the process of mitosis. Now, to understand the difference between mitosis and meiosis, we kind of have to present a little bit about the whole life cycle of, let's say, an organism such as us, Homo sapiens. So consider for me for a minute just kind of this simple illustration. Let's say there is a man and a woman, all right? They love each other very much, and that man was basically formed when the man's dad and the man's mom, on some starry night, when the mood was just right, they conceived this this person here. Okay, let's imagine. So for that event to happen, this person right here, his dad and his mom had to have fertilization between a sperm and an egg. Well, the sperm carries 23 chromosomes, and the egg carries 23 chromosomes. And what happened is the sperm combined with the egg, and it formed this structure, which is called a zygote, which is the first cell that's going to have two sets of chromosomes now. So you can see here's our two sets, 23 and 23, for a total of 46. Well, from this one cell, the very first cell that's been formed from this fertilization event, from this one cell, it's going to divide and then divide and then divide again and divide again and divide again, and it's going to divide over the course of 9 months of pregnancy and then over the course of the life, and it's going to produce the organism or the human. Okay, so this is all done through the process of mitosis. This cell divides through mitosis over and 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 over again, and eventually, after a pretty extended amount of time, forms a lot of complex structures and organ systems and things like that to form the fully functional organism. And because of that, all of the cells in this person, like whether it's his eyes or his brain cells or skin cells or muscle cells or pancreas cells or liver cells or small intestine cells, any cell in this person's body is called a somatic cell. And it is an identical copy of the zygote, because that's what mitosis does. It makes identical copies of cells. So you could pick a cell anywhere, even though the cells look different. You know, a neuron looks totally different than a skin cell, and a skin cell looks totally different than a muscle cell. They still bear the same DNA. It's just each one of those cells, the reason why they're different is because they're making different genes more than others off of that code of DNA, but still all of those cells have the exact same DNA because they were all formed through this process known as mitosis. Okay, well, let's say that this man here, he meets a woman who also at one point in time was conceived from a sperm and an egg of a couple, and the sperm and the egg combined to form what was known as the zygote, and then from that zygote that now is called a diploid cell, so this would be called diploid, whereas each of these are the gametes, sperm and egg, are called haploid. They're called gametes. All right, so a haploid and a haploid cell, one set combines to form a zygote with two sets, and let's say that zygote over many months underwent a whole lot of mitotic divisions, and then finally created this lovely woman that we see here. All right, so I apologize for the traditional gender roles of these simple figures, but at any rate, it kind of helps to get the point home. All right, so we have a man and a woman. Most of their cells are diploid because all the cells except the gametes are going to be diploid, and they're going to be identical copies of one another in terms of the DNA. All right, so let's say that this is the scenario. Well, what happens, let's say these two folks here, they fall madly in love, and on some starry night when the mood is right, they conceive a child. Well, how this works is that the male and the female, the male is producing a lot of sperm as his gamete, and the female is producing a lot of eggs as her gamete. And in order for these two to have a couple, one of the many millions of sperm from this male has to meet one of the many hundreds of thousands of eggs from this female. And you know that this one's going to have 23 chromosomes and this one's going to have 23 chromosomes. And together they could produce a baby of some kind. All right? So maybe a baby girl, could be a baby boy, but in either case, a sperm has to meet an egg. Now, there's a couple things I want to point out. The first thing is that the process by which these gametes are made is called meiosis. Okay? So this is different than mitosis because in meiosis, recall that the zygote is undergoing all these divisions, and mitosis is the thing that creates identical copies of the cell. You start with a diploid cell and end up with a diploid cell that's an identical copy. Well, in meiosis, all of these cells that are being created are haploid cells. So this is kind of a little bit unique when we're talking about cell division. Basically, what's going on to make these cells is that a diploid cell is being converted into all of these, which are haploid. Okay, that is the process known as meiosis. And so that's the first thing I want you to see, is that most of the time cells are going to have 46 chromosomes, but in these two special instances, the gametes, so the sperm and the egg, they only have 23 chromosomes. So there has to be some process in play to reduce the number of chromosomes by half, to go from a diploid cell, you know, some somatic cell in the body, and bring it down to haploid cells. Well, this is done in a couple of special organs called the gonads. So in the testes of the male, all of the time there are what are called stem cells that are diploid, and they're going through this process of meiosis to create all of these haploid sperm. And then in the female, it happens a little bit differently, but it's the same story. The female has some stem cells which are undergoing the process of meiosis to create the haploid cells here. So that's where the importance of meiosis. Now, what's kind of interesting then is when this sperm and this egg combined, again it forms a zygote that has 46 chromosomes, and the whole process begins. Now, what's interesting above and beyond that is that, let's say that this sperm that was made by the man met this egg that was made by the woman and created baby number one. All right. But then let's say on another starry night when the mood is right and we got some nice smooth jazz music playing, another baby is conceived. Okay, so let's take another sperm that's made from the man, and that's going to combine with another, a different egg made by the woman. Well, when that happens, that's going to form baby number two. And as we all know, baby number two is going to be not an identical clone to baby number one. So that should tell you that the DNA that is in all of these cells is different. And the DNA that's in all of these cells is different, because if the DNA in all of these cells was the same, and the DNA in all of these cells was the same, when this sperm, met this egg, it would create baby number one, and when this sperm met a different egg, it would create a baby that's an identical clone, because all of the DNA is different. So that are kind of the two properties of meiosis. One, it creates haploid cells from diploid cells, so that's unique. And then the other thing is that meiosis is going to make genetically, I guess I'm not really sure how to say this, but genetically non-identical cells. Okay, those are kind of the two properties of meiosis. So let's go ahead and take a look at this process. We'll get away from our little stick figure man and woman figures. So the process of mitosis, just for reference, looks like this. The cells will spend a lot of their time in this thing called interphase. And then they, at some point in time, begin what's known as the M phase. And in the M phase here, we're talking about mitosis. And mitosis has prophase, prometaphase, metaphase, anaphase, and telophase, just like we talked about. Well, meiosis is a little bit different than that, because it actually has two steps to it. There's meiosis 1 and there's meiosis 2. And in meiosis 1, this is kind of the big interesting step where the cell goes from diploid to haploid. So basically, one diploid cell is converted to two haploid cells. And then in meiosis II, each haploid cell divides to form two haploid cells. So if you do the math here, if you have one diploid cell making two haploid cells in meiosis I, and then two haploid cells, each one of these makes two additional haploid cells, then we would come up with 4 total haploid cells at the end. So it looks something like this. The beginning, there is this cartoon is basically trying to show you that this cell is a diploid cell, and one of the ways that we say diploid is to write 2n. 2n refers to the fact that there's 2 sets of chromosomes. So anytime you see that word 2n, just think it means diploid, 2 sets of chromosomes. All right, and so you can see there's the set in red, and then there's the set in blue, and it could be that, say, like the red is the set of chromosomes that came from mom, and the blue is the set of chromosomes that came from dad. All right, so again, there was a zygote that was formed when the parents that made this cell conceived, and the sperm and their egg met. That zygote divided up gazillions of times to form the organism, and then at some place called the gonads of this organism, it takes one of these cells that's an identical copy from that original zygote cell, and then it starts to do something interesting with it to change it into cells that are not identical anymore. This is a process that has to be done to make gametes, and that's what meiosis is all about. Okay, so here we see 2 sets of chromosomes, a smaller one and a bigger one, on a smaller one and a bigger one. These 2 would be called homologous chromosomes. All right, so in prophase 1 of meiosis I, what's happening is that the chromosomes condense, and they're actually duplicated before during interphase, during the S phase, they're duplicated to form sister chromatids. And one thing that's interesting that happens in prophase 1 is that they are going to form this structure called a tetrad, which I'll show you in just a minute. Okay. And then there's a whole bunch of interesting stuff that's going on here, which we're going to talk about in a minute. But then just for now, I'll show you that, in the end, these are the 2 cells that are made at the end of meiosis I. Okay, and you can see that these, in fact, are haploid cells because they've got a duplicated big chromosome and a duplicated small chromosome, but there is only one set. I mean, the set is duplicated. There are sister chromatids here, but there is only one set of chromosomes, and there's only one set of chromosomes here. If there were 2 sets, then you would expect to see another pair of sister chromatids that are this size, and then another pair of sister chromatids that are this size. Those would also be in the nucleus as well, but that's not the case. So we've gone from diploid to haploid in meiosis I. Okay, then in meiosis II is very similar to mitosis. In the way that the chromosomes arrange and are duplicated. It looks something like this. The chromosomes are duplicated, sister chromatids, just kind of like how they are here. Okay, and then they line up in a single line just like they do in mitosis. Then the sister chromatids are pulled apart, and you can see that each cell makes 2 new cells. Well, we're starting with 2 cells. So because each cell makes 2 cells, we have a total of 4 cells made from one original diploid cell in the process of meiosis. Now, one of the things that I want to point out, so we talked about there's 2 features to meiosis. It makes haploid cells, but it also makes genetically non-identical cells. So when you look at these cells that are made in mitosis, you can see that if you look at the DNA, you have a whole set of dad's chromosomes, a whole set of mom's chromosomes, and those are identical to the original cell. And you have a whole set of dad's chromosomes and a whole set of mom's chromosomes. Those are held in maintained from the original cell. But if you look and you zoom in here, you can see not only is there just one set now, but if we get really close and we look, you're going to see that the DNA has kind of been changed a little bit. Not only is it just a solid blue chromosome, it's a solid blue chromosome with a little bit of red on it. And this one's mostly red with a little blue. And then some cells have dad's smaller chromosome. Other cells have mom's smaller chromosome. And some cells have mom's bigger chromosome. And other cells have dad's bigger chromosome. All of the chromosomes that came from that original zygote have been flip-flopped and switched around, and they've changed DNA and genes, and then they've kind of randomly packaged up into these cells, which are now going to be matured to form what are called the gametes. And so you can see that they are in fact haploid. They only have one set of chromosomes. Here there are two sets, because these two are the same homologous chromosomes and these two are homologous chromosomes, but here we don't have that. And that's because they're haploid, and then also they're genetically non-identical. Okay? And that's important, because when I went back to this big figure up here, and I said that if this gamete meets this, it forms one baby. If this gamete meets this egg, it forms another baby. Those babies aren't identical because of the very fact that all of these gametes are not identical. The process of meiosis ensures it. It makes it so that all the gametes are different from one another. Okay, and that's good. Otherwise, we'd have a whole bunch of clones walking around the Earth, and there would be no diversity out there. So that's the process of meiosis. Now, the one thing that I want to spend most of my time talking about here is how do we achieve these two features? How do we, maybe you caught it, maybe you didn't, how did we get to a haploid cell? And then how do we get these genetically non-identical cells? That's totally counter to the process of mitosis. So what are the processes driving that? Well, to answer the first question, how we went from diploid to haploid, I'll show you the kind of the sneaky reason here. In metaphase 1, if you compare that to metaphase of mitosis, you can see that in metaphase of mitosis, the homologous chromosomes all line up in a single line. Okay? Well, a key difference in meiosis is that in metaphase 1 of meiosis 1, they don't line up in a single line, but they rather line up side by side, the homologous chromosomes. So you can see in mitosis, all the homologous chromosomes, because they're lined up, when they get pulled apart, it forms two cells with exactly the same number. But when you have the homologous chromosomes lined up side by side, when they get pulled apart, the homologous pairs are actually getting pulled apart. Here the sister chromatids, the duplicated sister chromatids, are being pulled apart, but here it's the actual homologous chromosomes that are being pulled apart. And so this is the step right here where the cell goes from being diploid to forming two haploid cells. Because this homologous chromosome, let's say from dad, goes this way, and this homologous chromosome that originally came from mom goes this way. This one from mom goes this way, and this one from dad goes this way. So you're splitting the two sets into individual sets on either end of the cell. Okay, so now, at this point in time, this is haploid. So that is what answers the story of how these cells become haploid. It's really this step here, metaphase 1, because they're lining up side by side, not in a single line. Now, the second question about why these cells are non-identical, there's a couple of different reasons. The first one is that depending on which chromosome lines up side by side, it's going to, when they pull apart, it's going to be really different results. So like in this picture, you're seeing that the blue one is on this side and the red one is on this side. So when these and then down over here, the red one is on this side, but the blue one is over on this side. So when these pull apart, this one, forms the red and the red bigger chromosome and the blue bigger chromosome and the red smaller chromosome get packaged up into one cell, and then the red bigger chromosome and the blue smaller chromosome get packaged up into a different cell. Well, this could not. This could also happen a different way. It could be that rather than the blue of the big chromosome lining up on this side and the blue of the small chromosome lining up on this side, could be that both the blue chromosomes lined up on this side and both the red chromosomes lined up on this side. Well, if that was the case, it would create two different types of cells where all of these had blue chromosomes and all of these had red chromosomes, meaning these two cells that were formed, one of them would have all of the DNA that came from this person's dad, and the other one would have all of the DNA that came from this person's mom. So it just varies, and this is what's known as the law of independent assortment. That and this is kind of the first reason why you and your sibling, if you have one, are not identical, because the sperm and the egg that were produced from your parents are all different. One of the reasons is because when they undergo that meiosis 1, and specifically during metaphase 1, where the chromosomes line up, it varies. So like here's a picture showing all the blues lining up on one side, the red lining up on the other side. This could also be a case where the blue and the red lined up on one side and the red and the other red and the blue lined up on the other side. And if you kind of just follow this picture, you can see what two cells these would yield. So this first combination, you're seeing it forms two cells that have all of, like, let's say dad's DNA, and two cells that have all of mom's DNA. But if they line up differently, then you'd have mixed mom and dad DNA between all the cells. And that's one of the reasons why, like, you might have traits that are like your mom, or you might have traits that were like your dad, depending on which of these pieces of DNA got packaged into your parents' sperm and egg when they conceived you. Okay. Now, in order to understand all of these processes, you do have to kind of think back like a generation or two, and just remember that in your DNA right now, it's not your DNA. I know you'd like to think it's your DNA, but in reality, all of your cells have half of your mom's DNA and half of your dad's DNA. That's what makes up your DNA. And then when you when gametes are being produced by you, it's taking the DNA that's from your mom, the DNA from your dad, and it's kind of swapping it around, and the sperm are going to have some of your dad's DNA or perhaps some of your mom's DNA. It just kind of varies. All right, so independent assortment, long-winded story. Chromosomes can line up on either side, and it makes a big difference. The next reason why there could be some genetic variation between these cells has to do with what's going on right here in prophase 1, and then also then in metaphase 1. Okay, what happens in prophase 1 is kind of an interesting thing. The homologous chromosomes are actually going to, through a process known as synapsis, bind together. And so one homologous chromosome binds to its corresponding pair, and they form the structure called a tetrad, which is basically just like a solid 4-chromosome structure, because keep in mind, each one of these homologous chromosomes has already undergone the S phase, so you can see there's, each has a sister chromatid. And so altogether, there are really 4 chromosomes here, or the potential for there to be 4 chromosomes here, and they're all fused together. Okay, so this is what's happened. This thing is being formed in prophase 1 of meiosis, and what happens is that that forms, and this picture is a little misleading because it shows that these are not still attached, but they probably would still be attached at this point in time as those tetrads. And here we're seeing, like, this is one tetrad, this is another tetrad. And in metaphase 1, there's this interesting process known as crossing over. And crossing over is where the homologous pairs are stuck together and they can actually swap DNA. So like when you see in this picture here, now this red chromosome suddenly has a little bit of the blue DNA on it, and this blue chromosome suddenly has a little bit of red DNA. So they've actually swapped genes at that locus, or at that spot on the chromosome. And so what happens in this is the two homologous chromosomes will line up, and let's say that there are a series of genes on these chromosomes. So let's say that this is dad's gene, and let's say this is dad's DNA, and let's say this is mom's DNA. Well, let's say these are all different genes. There's gene A, B, and C, and maybe this gene here is the gene for hair color, and maybe this gene here is like the gene for eye color. and then maybe this gene is like the gene for like the shape of the nose or something. Okay, and dad has what we'd call dominant genes, and mom has recessive genes, or it could be vice versa. We'll learn more about that here in the next chapter. But in either case, these two like complementary or not complementary, but these two regions of DNA that have the same types of genes on them, they can bind and swap DNA. Where they actually attach to one another is called this structure known as the chiasmata. And so these two chromosomes, one from mom and one from dad, are actually going to bind together and kind of cut off DNA from each other and flip it onto new chromosomes so that you get what's called recombinant DNA. So, or recombinant chromosomes. This word recombinant means basically that DNA has been switched or arranged somehow. Okay, so that's this process crossing over, and that's another reason why the gametes that are produced are genetically non-identical. Not only can there be variation in how they line up at the metaphase plate, but then in addition to that, there's variation in how they swap DNA. It happens kind of differently for every time that this process occurs. And so it's going to produce a bunch of sperm or a bunch of eggs that are genetically non-identical from one another. This is kind of a picture showing you a little bit of how that works. So in prometaphase 1 of meiosis 1, the two regions here are going to fuse together, forming these structures known as chiasmata, and then they'll swap DNA at that spot. And then whereas in meiosis 2, because there isn't two homologous pairs lining up side by side, there's just the duplicated sister chromatids, then this whole business about chiasmata does not happen. So all of this crossing over, this is going to be in kind of prometaphase and metaphase 1 of meiosis 1. Okay. All right, so there we have it. Meiosis makes 4 haploid cells that are non-identical. Mitosis makes 2 diploid cells that are genetically identical. And the place that we go from haploid to diploid is right here. And also the place where a lot of genetic diversity that we see is also happening kind of right here in meiosis 1 because of the law of independent assortment and because of the crossing over that occurs. Let's take a little time to unpack this. I've got a series of processes involved with the whole meiosis cell division, and I want you to see if you can match all of these up. So go ahead and pause the video and see if you can figure out where each of these descriptions, which one of these processes they match. And while you're doing this, just keep in mind that in meiosis 1, you have there's homologous chromosomes lining up side by side at the metaphase plate, and those homologous pairs are also having each chromosome in the pair has a duplicated, so it's got sister chromatids. And then in meiosis 2, it's a lot like mitosis, where there's still duplicated sister chromatids, but they're lining up now single file, so that when the sister chromatids are pulled apart, forming new chromosomes, it forms 2 new cells. So keep that in mind as you're working on this and see how far you get. I'll tell you what the answers are in just a minute. Okay, here we go. Looking right up here at A, individual chromosomes, here's kind of a tell here, arrive at separate poles of the cell and decondense. Well, if we know that DNA or nucleic material is moving to separate poles of the cell and decondensing and then dividing into 2, that's the process of telophase and cytokinesis. So then we just have to figure out if it's telophase 1 or if it's telophase 2. And because it's the individual chromosomes, that's going to be telophase 2. If it was telophase 1, what would be the thing that would be being pulled apart into different ends of the cell would actually be duplicated chromosomes, or another way to say it is sister chromatids. Okay. So then here B, spindle microtubules attached to fused kinetochores. Okay, we know that microtubules attached to kinetochores in the process of prometaphase. So then it's just a matter of if is it prometaphase 1 or is it prometaphase 2. And here they say that it's attaching two fused homologous chromosomes. So you have a tetrad, which is two homologous chromosomes bound together, and it's attaching onto that either side of that kinetochore, and it's going to pull apart those homologous chromosomes. So if that's the case, it's going to be prometaphase 1. Okay, the next one down, sister chromatids condense and the nuclear envelope begins to fragment or break down. Well, the condensing of DNA and the breakdown of the nuclear envelope, this is prophase. So then again, the trick is just determining if it's prophase 1 or if it's prophase 2. If it was prophase 1, it would actually be homologous chromosomes that were condensing down. And if it was prophase 2, it would just be, there wouldn't be homologous pairs, it would just be the individual duplicated sister chromatids. Now keep in mind, in prophase 1, there are sister chromatids present, so you could make the case that maybe this is referring to both of them, but I'll show you in a minute. There's another reason why this is not the case, because there's a better answer choice later that occupies prophase 2. Okay. So sister chromatids condense and the envelope breaks down, so that is going to be prophase 1. Or sorry, prophase 2. I might have misspoke a little bit earlier, but that's prophase 2. All right, homologous chromosomes randomly line up side by side at the metaphase plate. Okay, the key is that we have homologous chromosomes, homologous pairs, and they're lining up side by side, like I've drawn here. And if you have chromosomes lining up at the metaphase plate, that is going to be metaphase. So is it metaphase 1 or metaphase 2? Well, the answer is metaphase 1, because that's where homologous chromosomes are going to be incorporated. Sister chromatids are pulled apart, so imagine two sister chromatids and then pulling those two sister chromatids apart. That is going to be anaphase, and specifically anaphase 2, because if it was anaphase 1, the thing that's actually being pulled apart is not the sister chromatids, but it's the homologous pair of chromosomes. Chromosomes condense and homologous chromosomes bind firmly together. So this is kind of back to what I was alluding to over here. We have the condensing of DNA, so this has got to be prophase, and we know that we're talking about homologous chromosomes here. So this must be prophase 1. And so this one here then has to be by exclusion prophase 2, even though keep in mind in both prophase 1 and 2, sister chromatids are technically being condensed. Okay, down to G, sister chromatids arrive at different poles of the cell and the nuclear envelope reforms. This is telophase or telophase, and sister chromatids are the things that are arriving at different poles. Well, imagine a big cell, and I've got some sister, there's some sister chromatids arriving here. Here are the sister chromatids, and they've arrived there. Here are some sister chromatids that have arrived there. So this has to be telophase 1. If it was telophase 2, there wouldn't be sister chromatids arriving at different ends of the cell. It would actually be single chromosomes. As soon as the two sister chromatids break apart from one another, they're no longer called sister chromatids. They're called chromosomes. All right, on to clear up some space here. On to H. Spindle fibers attached to kinetochores of sister chromatids. So this is going to be prometaphase, and we've already done prometaphase 1, so the only other option here would be prometaphase 2. So imagine there are some sister chromatids in a cell, and the spindle fibers are attaching to the kinetochores on either sister chromatid. That's kind of the story that's happening in that description. Spindle microtubules pull homologous chromosomes apart. Anytime there is the pulling of something apart, that's anaphase. And we are told that the sister chromatids are still attached at the centromere. So the things that are really being pulled apart are the homologous pair of chromosomes. So that would be anaphase 1. And then lastly, sister chromatids are arranged in a single line along the metaphase plate. That is going to be metaphase 2. Okay, how'd you do? Were you able to get most of these? If so, that should suggest that you've got a pretty good understanding about kind of the ins and outs of this process. There's a nice table in your book that gives a little bit of a summary between these two in case you get a little confused. So DNA synthesis, where this happens in meiosis and mitosis, it's the same. It happens in S phase of interphase. Okay, so that's where all of the chromosomes are going to duplicate to form sister chromatids. The synapsis of homologous chromosomes, that is the fusion of those homologous chromosomes to form a tetrad, is what we'd call. This happens in prophase 1 of meiosis, and it does not happen in mitosis. There is now a tetrad formed in mitosis. Crossing over, this is going to happen during prophase 1, and it does not occur in mitosis. And this is a little bit confusing because they don't show this in this figure that we looked at. You know, if you look way back over here, this figure here, you don't see that crossing over has occurred by the time we reach prometaphase, so that's kind of a flaw in the book, I think. And crossing over does not occur in mitosis. Okay. Homologous chromosomes line up at the metaphase plate. This happens in metaphase 1, and it does not occur in mitosis. And they really should specify line up side by side, because homologous chromosomes do line up in mitosis. It's just they line up in a single line, not side by side. So I guess 2 bones that I have with this book on this topic. Sister chromatids line up at the metaphase plate. So in this, we're talking about individual sister chromatids like this, lining up in a line in the metaphase plate. So that happens during metaphase 2, and that happens just during regular metaphase of mitosis. So in that sense, remember that meiosis 2 and mitosis are pretty similar. And then lastly, this is kind of the important aspect of all of this. The final outcome is that meiosis will yield 4 haploid cells, and these are going to be not identical. And mitosis is going to make 2 diploid cells that are identical, genetically identical to one another. Okay, and if I gave you a little description about either one of these, you know, which process makes 4 haploid cells which are genetically non-identical, could you tell me if it was mitosis or meiosis? Or similarly, you might see something similar to this on a exam, hint, hint. So could you tell me a little about these and kind of do some of that sorting out of the different processes? Okay, one last note. I've already basically talked about this, so I'm not going to spend much time on it, but this just kind of illustrates what I was referring to earlier. In a male and a female are these organs called the gonads. It's the testes in the male and the ovary in the female, and this is the place where meiosis is occurring exclusively. And through that process, a diploid cell that is kind of taken, you know, there's a cell in the gonad that's a diploid cell, and it's committed to then going through this process of meiosis, and it will either, in the ovary it will form eggs, in the testes it forms sperm, and then these are both haploid, 2 haploid, then cells come together to form the diploid zygote, and then that's where mitosis is then going to take over. So meiosis makes the gametes form the zygote, and then from then on it's mostly mitosis creating diploid identical cells. Now the thing that tells a cell whether to undergo mitosis or whether to undergo meiosis, that's kind of lost on me. I'm not really sure exactly what all of those mechanisms are. but there must be some sort of chemical that's released or some sort of a protective environment in the testes or in the ovary that tells those cells specifically to do meiosis and not do mitosis. In fact, there is what's called the testis blood barrier. So there's a very isolated region in the testes that's kept separate from the rest of the body. And I think in that environment is where a lot of those chemicals are released to tell the cell to undergo meiosis. And that's important because generally it's a bad thing for cells to undergo meiosis. You wouldn't want all the cells in your body like being capricious, randomly doing mitosis and then randomly doing meiosis. It's important that all the cells that are supposed to be doing mitosis, they actually do mitosis, and only a select few cells called the gametes do meiosis. So that's how that's all regulated and controlled is kind of interesting, but I'm not exactly sure all of the mechanisms involved. That's going to wrap it up for the process of meiosis.